Can you guys hear me in the back pretty well? Yeah, we're all good? Cool. Yeah, so I'm Miles, my company is Terraforma. We are a soil biology and regenerative focused company. We tend to work as indicated with mid to large size growers, oftentimes conventional growers that are either looking to make a shift towards slightly more regenerative practices or going all the way to an organic or regenerative ecosystem for production. I don't have to spend much time on this slide. I had a great introduction. We don't have to look at the photo my wife photoshopped for me very long. We'll skip right over that. Just to kick off before we get into anything too technical, and we are going to try to get into the weeds a bit, so hopefully everybody's not too tired post-lunch. I just want to highlight some concepts around regenerative agriculture more generally that we hold as an institution, as a company, that we try to build into all of our projects. Those being growth over the long term, especially in the conventional agricultural system, we tend to see resources getting leveraged over short-term intervals to maximize yield for that given year. There's a big focus on immediate profitability and not a very large focus on long-term growth and sustainability. So trying to put a numerical value on the long-term benefit of taking care of soil, enhancing soil biology, and growing in a way where we're taking care of our environment as well as our soil health is something that can help to reprogram people's mindset around that initial short-term gain in favor of looking over a longer-term period. We also want to maximize benefit. This comes across the board. We do work, again, with a lot of these guys that are still using fertilizers. So we want to make sure that we're using every ounce of that fertilizer that we do use and gets into the plant. Same goes for our soil. We don't want to be wasting soil resources. We have this incredible network of minerals and of microorganisms that live within our soil ecosystem. And if we're not leveraging that to grow our plants, we are wasting that valuable resource. And often people go too far one way or the other, where we're super reliant on fertilizer and we're not leveraging any of that native microbiome to do any of that heavy lifting for us. Or we're not using any of those tools, and sometimes in organic production, we're left not able to produce very much produce. And that's where sometimes these mixed regenerative programs can be more productive. Reducing waste is a big one. This is mostly in concern to the environmental impact of waste, runoff from nitrates into the water supplies. Wasting chemicals is money in your pocket as well. And we want to reduce the amount of pollution and waste that we see within our systems as much as possible. And then, of course, building self-reliance. This is trying to create systems that can defend themselves, that can produce for themselves, that have the capacity to be able to build some of their own value within the landscape, as opposed to having to generate all of that value externally and spoon-feeding crops consistently. And then finally, we do want to see environmental benefits. But we want to move that needle in a way where we're coming through profitable mechanisms. We hold really core to our belief system that if we want to make an impact on a really large scale, the only way we're going to start to move those practices in a meaningful way is if big institutions see benefit in environmental restoration. And at the end of the day, no matter what we do, that comes down to dollars and cents for them. So I want to start by diving into a concept called succession. This is effectively the building of complexity within an ecosystem. So starting from our most basic landscape, where we're just dealing with minerals, it's basically a sandbox, like a beach. Nothing's capable of growing there. So how do we get from this very early stage in succession to these really complex forests and jungles and rainforests that we might see in South America? It's a slow process driven predominantly by the microbial inoculation of that soil and the increasing divergence from bacterial to fungal biomass over time. This process is also driven by the plants that are seeded into that ground. One of the things you'll notice in a very early ecosystem is you tend to get a lot of weed pressure, right? It's a lot of quick annual growing weeds, and that is because they're able to leverage those resources in an effective way. There's not a lot of organic matter. There's not a lot of solubilization of nutrition taking place. Those seeds come prepared with enough nutrients to be able to land in the soil and grow very quickly. And they actually serve an ecological function for us as well. When we're seeing that kind of heavy weed pressure, they're attempting to build organic matter in that system and make it palatable for this next series of plants to be able to take hold. Why do we want to see this increasing amount of fungal diversity within the soil as we get into higher complexity systems? This is largely driven by the capacity of fungi to be able to create networks within the root systems, but also to acidify soil and to produce more ammonium over nitrate. When we talk about nitrogen, there's two different forms, chemically speaking. We have ammonium, which is going to be more stable in soil. It's not going to leach out through excess water, through rain events very easily. And we have nitrate, which is very easily leveraged by plants for rapid growth, but it's also very soluble with water. So that is typically how we're leaching a lot of that nitrogen from our soil systems into our waterways. These fungal ecosystems produce predominantly ammonium, and they tend to produce a lot of organic acids. Those two things combined lower our soil pH, and they're able to support slower growing plants like trees in these really advanced ecosystems. So when we're looking at any soil samples that you might find from an older growth forest, you're typically going to see like a pH 5.5, heavily fungally dominant soil. On the other end of that spectrum, we're going to see a lot of bacteria, right? What do bacteria do in soil? They tend to make soils more alkaline. They operate around like a 7, 8.5 pH, and they tend to produce quite a lot of nitrate. So if we're dealing with a soil that's producing a lot of weed pressure and is very susceptible to loss of nitrates, it's usually very bacterially dominated. And when we're in an agricultural system, we do need some amount of both of these things. If we want to produce corn and vegetables, we're going to want to have a roughly 1-to-1 bacterial and fungal ratio to be able to leverage some of those nitrates that are going to come from the bacteria for fast growth of these heavily vegetative plants, as well as have that dominant fungal biomass to be able to produce that ammonium within the soil. If we're working with orchards, we're going to want to push that farther into the fungal realm. And by controlling some of those balances within the ecosystem, we can support more complex life, or we can produce more heavy weed pressure. The next thing we're going to talk about a bit is how we destroy this system. We're going to cover that in a bit of detail of these different practices that are going to create negative impacts on successional stages, and how we continuously push ourselves farther and farther away from ecological complexity towards simplicity within most of our traditional agricultural methodologies. The practices that are going to be highlighted here include tilling, fertilizer, fungicides, herbicides. Obviously, burning in your fields is not a good idea either. Generally, to start with tilling, we've talked a lot about this throughout the past 2 days, I think, so I'm not going to beat it to death. But when you do till, most of our organisms live in this top 6 inches of soil. And so when you till that under the ground, you're disturbing all of that microbial activity. You're pushing it underneath the surface. You're also increasing the amount of oxygen that's going into the soil. And at first thought, that might be a good thing. Oxygen in the soil is good. It's good for microbes. It's good for plant growth. However, it creates bacterial blooms within that soil ecosystem. That increase of organic matter and then the contact within the soil media and then all of that oxygen activates bacteria to start volatilizing that organic matter very, very quickly, and it's going to push a heavily bacterially dominant soil very rapidly. You're also destroying fungal networks. They grow for miles under the surface of the soil in some instances. Largest organism in the world is a fungus that grows underneath a forest in Washington. And so when you rip that apart, you're killing those networks of fungal hyphae that help to connect all of these plants, to solubilize nutrients, to move water within the soil from one plant to another. You're also going to be increasing compaction at the point that your tilling machine reaches. So you're only loosening soil to the point that your blades go deep into the soil. If you till it, say, 12 inches, you'd have a new compaction pan at 12 inches. And that's going to be a source of runoff from your soil. You're going to see water moving at that compaction point, and it's going to be very difficult to break that up over time. And that is where the practice of no-till, the capacity to produce and leverage the tools that we have available without tilling, can build up soil health and structure over a longer period of time. When we talk about fertilizer, this is something that a lot of regenerative growers try to stay away from quite a lot. We have seen instances with clients that we've worked with where utilizing fertilizer can be helpful. We've seen it do very bad things. It really comes down to quantity at the end of the day. We systemically over-apply nitrogen and phosphorus and a lot of these types of fertilizers, NPK, and that leads to this excessive runoff. That leads to root structures that aren't forming associations with microbes and weakening the plant. That's leading to high salinity in soils, and microbes do not love salinity in soil. They are very weak to changes in salt composition. So we want to make sure that we try to use the correct amount of fertilizer when we are using fertilizer. We want to maximize that benefit as much as possible. We also see increases in denitrification. We'll touch on that. We'll talk about the whole nitrogen cycle from a microbial perspective in a couple of slides. But it's going to increase that loss and runoff. It's going to shift your microbial community significantly. So we want to make sure if we do use fertilizer, we're not overdoing it. Herbicides are going to be one of the things that we try to limit the most when we're working with clients and working in fields. They not only damage the weed that you're trying to kill, they're going to damage your cash crop as well. We have a lot of genetic resistance that we've bred into different plants to be able to resist these herbicides. It still hurts them, right? If you dig up your root system after you do an application, most of the time your roots are going to be black. So you are detracting from your ability, the plant's ability to uptake nutrients to be able to function properly within the soil, even though it may not show a complete death symptom. You are also going to be killing off a lot of the bacteria, a lot of these other organisms within the soil. Herbicides are not really made to just kill plants. They have run-on effects within that ecosystem as well. And this is going to be generally a running theme throughout most of the chemicals that we use. And then, of course, these chemicals can be damaging to human health. When we spray them, we consume them, it's going to have an effect on us. Farmers are the most exposed to these chemicals typically because they handle them. So from the outset, reducing the amount of people that get exposed on the job is going to be a huge benefit for families that work within the agricultural industry. Fungicides, yet another chemical that does not have a targeted way to kill just the fungus you want to kill. When you apply these in the soil, you are killing a whole bunch of fungus. It's all the beneficials as well. And once we get in a system where you've knocked out all of your beneficial fungal biomass or your beneficial bacterial biomass, the pathogens move a lot faster. Their morphology and the way they grow is to colonize quickly. They don't grow mushrooms in reproductive bodies. They explode into the soil looking for food sources. And they're going to move a lot quicker than any of the beneficial biology that can recolonize those soils. The second, I know a number of people have talked about VAM and ectomycorrhizae. They are incredibly important organisms to associate with plants. Fungicides do have an effect on these microbes. Tilling has a massive effect on these microbes. We want to make sure we're preserving those communities and building that relationship with plants to help solubilize things like phosphorus, right? If we have these mycorrhizae in place, we're capable of reducing our phosphorus usage within the fields as well. So all of these things end up tying together. When we look at pesticides, this is another one where we have systemic problems. You apply a pesticide, yes, you're killing your target pest. Sometimes they can be relatively selective. Usually you're killing all of the insects in the field. And yet another one where if you don't have predatory insects in your fields, predatory insects in buffer zones, you're not going to have anything to stop them from coming back into those fields. You're also killing lower level microbes as well that you might not see with your naked eye. There are insects in the soil that enact critical functions that also get killed by insecticide. There are a lot of farms and fields that we've gone into and you cannot hear a thing. There's not a cricket, not an insect buzz, absolutely nothing. When you walk into a healthy field where you're recultivating soil life and an ecosystem, you will hear the buzzing of insects and the chirping of a lot of different species within those areas. All right. So in the end of the process, when we've implemented tilling and herbicides and different chemicals within our systems, we're leading to an entire ecosystem collapse. And it happens rather quickly. You might not lose all of your organic matter year one. It might take some time. However, you are breaking down what would be soil structure through the integration of hyphae and insects and earthworms, and you're creating compacted zones. Every time we till, it just compacts a little bit faster each time. And so all that air that you're creating is disappearing rather quickly. We want to, as much as possible, maintain this structure, maintain that organic matter into the future, and build those communities. Without them, we're going to lower our CEC, which is our cation exchange capacity, which is basically how much nutrients can my soil hold? How big is my cup? And if we're working within a chemical system, if your cup gets smaller, but you keep applying 200 pounds of nitrogen every year, less of that is getting held in that system. So we want to make sure we're continuing to make that cup bigger year after year so we can hold more of that nutrition within that soil profile. All right, moving into the part where we talk about the good things and how this all kind of functions and how biology can help to make nutrients more available for plants, as well as replace some of those traditional methods of fertilizing plants. It all starts with the plant itself. The plant is going to effectively photosynthesize energy. It's going to produce carbohydrates and sugars, and about 70% of those carbohydrates and sugars are going to move into the root system and get secreted as exudates into the soil. Those are basically just saline molecules to microorganisms to say, hey, I'm here, I'm alive, come help me, right? Plants don't have the capacity to produce their own minerals. They can't produce their own nitrogen most of the time, pretty much ever. They're going to have to associate with a microbe to be able to make that happen. So they're calls for help by the plant, effectively, and they put an enormous amount of investment of energy in order to grow out that association with these microorganisms. The other half of the microbiome kind of dependent biomass is the organic matter. This is going to be composed of all of the leaf litter that grows above ground. It's going to be composed of dead microorganisms within the soil profile. This is another place microbes call home. They're able to break that down, grow off of that biomass. It's a class of organism, saprophytic microbes effectively. Fungi, bacteria, which are this first little class of organism here, are able to colonize that organic matter and begin to slowly break down those nutrients and incorporate that into the biomass that's living within that ecosystem. However, if we don't have these other microbes within that mixture, if we go up a trophic level, if we're looking at protozoa, nematodes, and arthropods, those are the organisms that actually eat the bacteria and fungi. They not only predate on pathogenic organisms that live in the soil, they're critical for making those nutrients available to the plant. If we just have a bunch of bacteria and fungi that are operating within the rhizosphere, supporting that plant, like eating the organic matter, their main goal isn't to keep that plant alive per se. Their main goal is to replicate, is to grow more of me. And so we need microbes in the soil that are going to say, hey, there's a bunch of fungi and bacterial biomass here. Why don't we come eat that? And when they eat that, that's when you start to release the nutrition. So a soil that only has fungi and bacteria is not a functional soil, right? It's only got balance from a single perspective. We need to go up the food chain a couple levels. And then obviously mineralization and solubilization becomes critically important. When we're looking at soil surfaces like soil colloids, heavy clay soils tend to absorb nutrients very heavily. Phosphorus in particular can be really hard to get off of those surfaces. And the only way that we can really work within the microbiome to get that off is to leverage fungi and bacteria that can locally change the pH within those different areas in the soil, leverage that resource, and then start moving it within the biomass of that system. Without that, you're going to have those nutrients locked up for quite a long time. So we're going to dive in a little bit now into the nitrogen cycle. This is effectively how the microbiome moves nitrogen within an ecosystem. This is important because it doesn't matter if you're using fertilizer or however you're putting nutrition in the ground. This is how it gets to plants at the end of the day, unless you're just putting pure nitrates in. So I just want to define a couple of these terms, starting with denitrification. This is a process where nitrogen is volatilized out of soil. It's a loss of nitrogen from the system. These are organisms, particularly bacteria, that are taking nitrate and turning it into N2 gas. And so it's just being lost and back into the atmosphere. This is one of the primary pathways where we're losing a lot of that nitrogen fertilizer. Most people think of it going through the ground. A lot of the time it goes back up into the sky, which is just a waste of money at the end of the day. Nitrification, this is a process that's critical to soil function. It takes ammonium, which is that stable form of nitrogen we had talked about, and it turns it into nitrate, which is that less stable form. That's important for growing plants. There's parts in a plant's life cycle, in the vegetative part of a plant's life cycle, it needs nitrate to grow those leaves. It needs nitrates to grow healthily, but it's another pathway for loss. If this portion of the microbiome is overregulated, if it's overactive, it's going to take all of that ammonium, shift it into nitrate very quickly, and that's when you start to lose it through the soil profile very quickly as well. So as much as possible, if we're trying to maintain nitrogen within our ecosystem, we want to reduce the amount of denitrification and nitrification taking place within the soil. In order to save nitrogen in the soil, or build nitrogen in the soil from one year to the next, we want to look into ammonification. This is the opposite of nitrification. It's taking nitrates, turning them into ammonium. So taking a less stable source of nitrogen in the soil, creating something much more stable in the soil that's not going to wash out with rain. It's a resource that can be leveraged throughout a growing season. We also want to encourage nitrogen fixation. This is something you'll see a lot with cover plants. Legumes are going to fix nitrogen within that system, add that back in. But also there are free-living nitrogen-fixing bacteria within the soil ecosystem. And they're capable of uptaking a lot of that N2 gas, putting it into their biomass, and then allowing that to be cycled back within that ecosystem for plant uptake. These can be very efficient microbes, but if you put a lot of fertilizer in the ground, they're going to shut down. They have external sensors. They know when there's excess nitrogen in the soil, and they will stop fixing nitrogen when they're surrounded by a bunch of soluble nitrogen within their ecosystem. And finally, microbes are actually capable of incorporating nitrates and ammonium and proteins into them. So if you are applying a soluble fertilizer into the ground, a healthy microbiome can help to hold that in the soil structure. They'll absorb some of that, and they'll be able to recycle that moving forward into your season. So if we can build out systems where we have a healthy microbiome within the soil, we're going to be capable of retaining more of that nutrition that would otherwise run off of these systems. So we can reduce the amount of fertilizer we use because we're going to leverage more of it and keep it within the soil profile. The question, though, is how much of that actually stays. This is a study that was done back in 2010 where scientists took external nitrogen, synthetic nitrogen, they put it into an ecosystem, and they tracked how that nitrogen moved throughout the soil and the plant. The main thing that I want to highlight here is you will see increases in the nitrogen in the leaf structure. You'll see increases in nitrogen above ground. You'll see increases in nitrogen in the leaf litter in the soil, the O horizon. However, you'll see a decrease in nitrogen in the biomass. You'll see massive increases in denitrification. You'll see almost 500% increases in leaching. So the amount of actual fertilizer that the soil's capable of utilizing within an ecosystem is not that large. If we're applying 200 pounds within a system, we're stimulating a whole bunch of this loss here, right? However, there is clearly a little bit of a benefit. We do see this absorption rate, this uptick within that system. We do work with clients sometimes where we remove nitrogen entirely from that system. We go completely organic overnight, and you will have problems growing enough biomass in some instances. And that will have a more detrimental impact on the microbiome than the fertilizer has against a control. So we need to have the biomass available to grow out a microbiome, and it's important that we can maintain that. So the question is, what's doing more harm, right? Is it more harmful that I'm reducing the productivity of this field, or is it more harmful that I put a little bit of fertilizer down? The next topic I want to cover here is diversity. This is a really interesting chunk of soil microbiology that I really encourage people to dig their hands into a bit. It's a little bit of a newer concept. People have focused a really long time on individual microorganisms, the effect of a nitrogen fixer, the effect of like a trichoderma or something like that to reduce pathogens. But there is an incredible capacity of the soil microbiome to be able to resist changes in their environment, to be able to recover from stress events if we have a broad diversity of microorganisms. That is because we have a lot of redundancy built into those systems. So we have organisms that serve the exact same function. We might have 10 different nitrogen fixers in the soil, but they work at different pH ranges. They work at different temperatures. They work at different levels of organic matter within the soil, different oxygen levels. So we want to make sure that we're stimulating a broad diversity of these microorganisms within an ecosystem so that when we do have a stress event, if we have drought, if we have too much water, that system can then recover effectively. There's also another concept called quorum sensing. This is effectively when microorganisms, the sum of the parts is, no, the whole is greater than the sum of the parts. Get my idioms right. Basically, when microbes interact together, they're able to change the function of those other microbes. So a community of people that are working together might be able to produce something greater than any individual of that group would have been able to create. And that is true within microbes as well. The signals they send each other, the impact they have on each other will increase not only the complexity of the soil, but what they can produce out of that soil for the plant as well. This resistivity factor is another one. This is effectively just saying something similar to redundancy, but we want to be able to have soil ecosystems that have a diversity of microbes to compete and reduce the amount of pathogens and problems in our soil. We want to solubilize as effectively as possible. And then competition is a huge one. There are billions of little pockets in all of our soil. If we're testing, and we'll talk about soil testing in a minute, if you test soil here and you walk 5 feet that way and you test it there, you can end up with very different results on a genetic level. So we want to make sure that we have a diversity of microbes in the soil so they can occupy each of those little niches that best fits them. And if they do that, they're going to be able to leverage those resources effectively, help make that available for the plant, and more importantly, keep pathogens out of that area as much as possible. This is another study done in 2007 that's just exploring the dynamics of ecosystems and growth systems. The main thing that I want to point out here, these little dashed lines indicate practices that are causing harm within this system. So you'll see things like tillage, pesticides, and their effect on soil biology, their effect on some pests. But the main thing I want to point out is where we find soil chemical and physical fertility in this diagram and where we find the beneficial soil biology, right? The interaction between these different dynamics is very much so driven by our soil microbiology. The structure in the soil, the chemical availability of different nutrients in our soil is driven by and large by the biology that live within our soil. So one of the things that I want people to start to think about more, and we always try to encourage, is think of your soil not as soil physical and chemical characteristics, but think of it as biological, physical, and chemical characteristics. Too often we let that slide by the wayside, but it's responsible for control of pests and diseases. It's responsible for enhancing the chemical and physical fertility of the soil. It's responsible for stimulating plant growth and stimulating the rhizosphere as well. When it comes to pest disease and occlusion, the main thing I want to hammer home here is this predation aspect, this occlusion and competition aspect that we've talked about a little bit, are incredibly important. This is how nature controls pests and disease. There's something that eats it. There's something that competes with it for those resources. If we can inoculate plants, build a healthy rhizosphere that's inoculated with beneficial microbiology, it's going to keep out the pathogens that want to attack that plant. It's a really effective way to guard plants against disease. And then biocontrols can also be utilized over traditional pesticides or in tandem with them. It is a little bit of a kettle, what is it, pot calling the kettle black, something along those lines. It's the same mentality of using a pesticide, right? It is not a one target, one shoe fits all solution, but it is healthier, right? You can rub your arms in trichoderma. It's not gonna hurt you. It will only hurt fungus. And they tend to be less devastating to ecosystems. They will recover, they can exist within them. Something like a trichoderma will inoculate, it'll kill big swaths, like large patches of organisms, which are usually disease causing because they tend to grow in a dense network. So they have evolved evolutionarily to deal with these types of pathogenic organisms. But it is a good tool to have in the toolkit. And then the last thing I want to leave here is just to remember that pathogens are opportunistic bad guys. We only call them pathogens because they do things we don't like, right? They are vital parts of a soil ecosystem. So the goal is not to eliminate them, it's to control them, to keep them in their own little corner, right? A small amount of pathogen will stimulate immune responses from plants. It's going to upregulate the production of compounds within the plant vitality of the immune system that are going to help defend against that disease moving forward. It's basically an indicator to say, hey, this disease is here, let's protect ourselves. So we don't want to eliminate them, we just want to make sure there's competition to keep them in check. Moving into soil testing, this is a really interesting and fun field in the microbial space and agricultural space that's coming together rather quickly. Doing soil analytics at the level of understanding the balance of the microbiome, understanding what microbes are present, can make guiding the solutions we want to implement more effective. If we can see that we're way out of whack on our fungal to bacterial ratio, if we can see that we're missing large swaths of predators within that system, we can then come in and we can address those problems with specific interventions. So it doesn't matter which analysis you use really, they all have downsides, like PFLA, which is a fatty acid analysis, is gonna have some shortcomings, right? Genetics analysis that use PCR, they're gonna have some shortcomings. So whatever you're using, as long as you're consistent with that usage, you'll be able to track progress and changes from practices over time, which is really important because, one second. It's very important because you want to be able to understand how practice changes are affecting the soil microbiome as well. There was a question in the back there.
Analysis is by using microscopic analysis?
There are a lot of different analyses out there. We usually do a genetics analysis combined with a direct microscopy analysis. There are a bunch of different labs that do both of those things. Like Biomakers, Trace Genomics, they're both biology labs that'll do full spectrum soil analysis through PCR. Mycorrhizal is direct through microscope. You can do those soil dyes. We actually have we taught a lot of clients how to do that themselves. If you have a microscope and you're interested, you can learn to do all of this. It's not super complicated. It's just a little bit of work and time. But you can do a lot of that yourself. There are more expensive options, there are cheaper options. There's a lot of handheld devices that work on farm that are like $5, $10 a sample. They're not as profound. However, they can still guide practices and usage and changes within your fields. Just be consistent with what you use. You cannot do like a PFLA and then go to genetics and expect to map those together. They're completely different. So just be fairly consistent with what you're using. And then on that decision-making side, just understanding, okay, now I have this information. How do I then turn around and make improvements or changes to this? Part of that is experimentation on all of your guys' part, right? I'm gonna try something different and see how it goes. And then the other half of that is learning how to implement solutions that are gonna be effective within those fields. Generally speaking, one of the great ways to do this is to increase the diversity through products. Like microbial mixes, basically. We've heard a lot about vermicompost from Nicole. We've heard a lot about different microbial stimulants and stuff over the past couple days. Those are all fantastic ways to increase biodiversity within the system. The one caveat to that, use local and native microbes. The idea of putting things in bottles and shipping them from California to New York, for example, is a bad idea. These microbes are quite like us. They have their own immune system. They have their own immunity. They grow in an area with an ecosystem, a temperature, an elevation that they're used to. If you try to ship those somewhere else and make them work, they're not going to be adapted to that environment. So it's important that you use things that are localized to where you are. The other half of that is, again, bottled. Anything bottled is going to be dormant. It's not going to be active. It's not going to be alive. It can't be reactivated very effectively because it's going to be a competitive mixture. So typically, it's recommended to stay away from bottled products as much as you can unless it's like a pure isolate of a single microbe. And we can cover that a little more if you guys are curious about like what products work kind of best and how to stimulate those and work with them. Mycorrhizae are incredibly important. Getting those implemented into the fields and associated with plants is a huge win in any system. You can do this yourself. You don't need to buy a product off a shelf. You can create your own mycorrhizae from ecosystems within your area. Again, not incredibly complicated. You can grow them in trash bags with a host plant, right? And then you can spread that across your fields. We've worked with growers that we've gone as far as to build hydroponic systems where we're producing like thousands of different like spores every day on like a 3-hour cycle. It's a bit more intensive, but it's possible to produce these things on your own farm. You don't have to necessarily go searching, and you have the benefit of better genetic fit for your biome, which is critical. Some of the other practices that we like to implement, we've heard a lot about cover cropping. This is huge when you think of that process of taking photosynthetic potential, turning that into sugars and turning that into carbohydrates and sending it into the soil to stimulate microbial growth. If we have fallow soil, that is what we're missing out on. We're missing the capacity of these plants to produce food for microbes within that ecosystem. We're missing out on the opportunity to be able to harvest sun for X amount of months of the year that you're in a fallow period, right? So we want to take advantage of these resources that just exist. It takes very little work for us to let plants photosynthesize at the end of the day. So we want to encourage that as much as possible. But you're going to be able to both protect the soil with soil cover, right? You're not going to have as much erosion potential. You're going to be able to enhance soil fertility. You're going to be pulling up nutrients that potentially would have washed away through erosion or through runoff. You're going to store them in trap plants over the season, and that slowly breaks down and becomes available over the next season. You can use things like nitrogen fixers to add nitrogen to the soil in an off season, and you can help to build up wildlife habitat through that period. This is a critically important function of cover plants, is keeping predatory species alive, creating habitats for insects that create benefit within our systems. Another practice we can run is a living mulch. We define this a little bit differently than a cover plant because we're talking about planting something in association with your cash crop. This might be like a clover under a peach tree, right? This might be planting corn stripped with alfalfa or something along those lines, right? Where you have these overlapping beneficial plants that can work together and that can build off of each other to enhance the productivity of both of those plants. Being able to find ways to incorporate these types of practices will help to build synergies between the plants, put more nutrition, make it more available for your cash crop, as well as covering the soil and building more organic matter over time. It's also functional for housing predatory insects, like integrated pest management processes. All of that goes a really long way when you have the correct flowering plants or the correct source of pollen for these predatory species to live throughout the year and then predate insects that are going to cause problems throughout the growing season. So moving into some of our projects that we've worked with, I just want to highlight sort of how some of these changes take place. We take an approach that is very much so baby steps, experimentation. One of my core beliefs is that all farmers are citizen scientists at the end of the day. If you're not having an innate sense of curiosity, if you're not trying new things, you're never going to change. And farming is one of those industries where you have an opportunity every single year to try something new and keep what works and change what doesn't. So we encourage taking smaller lots, smaller areas, trying something, seeing what evolves out of that, and then if it works well, you can expand that process. If it doesn't work well, you can start to kind of tweak it or change it or abandon a practice that doesn't work well for you. Most of these processes, we're starting with conventional farms. These are farms that are fungicide, herbicide, spraying all the time, fungicides every 2 weeks type of applications. This is a grape farm in Peru. They have about 95% sandy soil. They're in a desert, but they have access to water because they have a huge river that runs off the mountains. So they're able to grow things like grapes with 200 millimeters of rain every year. Some of the first things that we were able, I just want to point out, like this swap that we did from a fully conventional to organic system was not only much more productive, we were able to get about 30% yield increase over historical yields in a conventional system, but we also increased their profitability massively. So their costs went down and their productivity went up, transitioning from conventional to organic through a biological system. This is how much of their farm looked when we first got there. You can see mostly this under story here. This is all just weed pressure they used to spray herbicide for. You can see how sandy the like pathways are where they drive their tractors, do their sprays, et cetera. What we tried to do to treat this initially was transition away from herbicide and find a way to occlude the system from light. So we started using a mulching process. We started doing microbial extractions, getting them underneath the mulch to help inoculate biology back into that system. And then occlude weeds by just having like 3 inches of, this is sugarcane fiber that we covered the planting row with. You'll also see some of the biology returning, right? We start to see mushrooms coming back into the system. This worked, but it was expensive and it was time consuming. We had to keep importing all of this organic matter from outside of the farm into the farm. So we eventually transitioned into doing the associative cover plants. So you'll see underneath the canopy here, we've got Crotalaria, which is Crotalaria, which is sun hemp, basically. And it's a nitrogen fixing species. So we would come in after pruning the grapes and we would plant the nitrogen fixer underneath the canopy, and it would get sunlight to grow before the grape canopy closed again. And then we were able to come back through, and eventually we used sheep, but we were able to come back through and cut the cover plant, release that nitrogen back into the soil as the grapes were starting to grow. And so you're able to kind of incorporate this multi-use tool. So not only were we stopping weed growth, we were also putting more nitrogen back into the system, and that was key for starting to move away from fertilizer inputs for them. Eventually we started doing mixes. So we did the sun hemp, we mixed in sweet potato as well. It was a really interesting mix actually. We had really good coverage of the planting rows. We also got nitrogen fixation, and the root systems looked incredible. They were just completely intertwined, but there was not a whole lot of competition between them. The sweet potato went a lot deeper than the grapes went, and the sun hemp is just a straight shot root, taproot, that's fixing about 70% of its own nitrogen. So they're not competing amongst each other all that much. And after you get through that initial cycle the first year, a lot of that organic matter, those nutrients stay within the soil and they're available for the next year. So a lot of the time, the harder implementation phase is year one, when you have extra plants to kind of grow, but you don't have the infrastructure necessarily within the soil to support that. We eventually ended up here where we started planting the roadways as well. We mostly did this because we wanted to be more sustainable minded and like optimize just the usage of green space, get as much in the ground as we could. But it ended up being a fantastic refuge for beneficial insects. So we had orthopods, we had ladybugs, we had predatory wasps that were all able to live within this center row and the understory that would eat mealybugs, and they would be eating other insects within the system. Some of the things that helped this farm in particular transition fully away from chemical usage. We were making vermicompost, we were making normal compost, we were making biological extracts as a side product to all of that. They were making their own fish hydrolysate on site. We had all of the cover plants in place. We were implementing some of those biological controls to help deal with some of the insect pests and pressures. And we also started an integrated pest management system where we were growing a predatory insect on the farm and releasing it into the farm when we had peak pressure. And then we started doing sap and bricks analysis as well to just try to chart and understand the nutrient usage within the plant so we could be more adaptive with different amendments that we might use and when to use them.
What is a sap?
Good question. A sap test is effectively a blood test for a plant. So instead of analyzing tissue, we're analyzing the intravenous liquid that runs through it. So everything that's running through the phloem and xylem and phloem, we're able to sample that from an old and a new leaf, and we're able to chart how those nutrients are moving within the plant and in that moment in time, basically. There are a number of labs that do that in the US as well. And it's a really interesting way to visualize nutrient usage within a plant. It's a little bit more reactive than tissue tests because tissue tests, you're looking a couple weeks in the past. With this, you're looking at today, right now. This is how those nutrients get used. So you can really chart and understand in any given moment how that functions.
Were you then doing reactive, like nutrient sprays?
On this project, On this project, we were doing mostly, if we needed like an organic mineral spray or something like that, we would usually do it. We weren't doing a ton of that here. We were mostly using like fish hydrolysates and stuff to get amino acids into the canopy. Their biggest issue was typically nitrogen most of the time. So we were trying to use compost and like fish hydrolysate and amino acid products to increase nitrogen content. Yeah.
Were there any studies done to see like the nutritional quality or quantity or flavor of the wine or the grapes and the product?
Yeah.
Does this affect anything?
So these were table grapes, so it wasn't for wine.
Okay.
They didn't do like a really in-depth, they didn't do like a amino acid profile on the grapes or anything like that. But productivity went up quite a lot, and like they grade grapes based on quality. It's usually based on damage more than it is based on like taste quality. So like if you have a little bit of scarring from a fungal infection or something like that, it goes to like a B grade. But their grading remained pretty high throughout the process. They did have one of the things that I always warn people about because it happened here. They were fully organic and they had weather conditions they had never had before. It was a lot wetter than normal. And there was a huge outbreak of fungal disease across all of Peru in that area in Piura. And that hit them really hard. And they did not have the management practices to be able to deal with it. Their clusters of grapes were too close together. And that is always a problem and a vulnerability. If you switch off of chemical and go to organic and you're not used to managing a crop in that way, one of the vulnerabilities you suffer is if you have something weird happen that year that you've never had before. It could be insect pressure, it could be disease pressure. It can really throw you for a loop. So we always recommend, even if you want to end up organic, in that transitional period, don't be afraid to keep some of those tools in your back pocket. We always recommend, instead of spraying conditionally every year as a preventive, do the work to get in the fields, evaluate if you have disease pressure in certain areas, and if you have to resort to using something chemical, it's better to keep your farm like functional and alive than like flush $2.5 million down the drain because you didn't want to use a fungicide. You gotta run your business first, right? Were there any other questions? We're gonna move on from the grapes here, but yeah.
The numbers you showed us a moment ago seem a little bit backwards from what I'm typically hearing about regenerative agriculture, where the yield may not go up very much, but the profit goes way up.
Yeah.
You had the opposite. The yield went way up and the profit hardly went up.
Yeah, well, they're still running by and large a monocrop, like kind of they're growing grapes in high density, right? They're not planting trees in the mix. They're not doing a full regenerative type of agriculture. This is conventional farming done with a biological sort of tint to it, an organic style to it, right? We're still high density here. This is 60 acres of grapes planted in pretty tight areas. So it's run like a conventional farm. They're trying to hit conventional metrics for production, and they're trying to maintain high profitability, like high revenue, as well as like finding that profit margin. The cost structure that shifted here, they ended up paying a lot more because prices for fertilizer were going up anyway, and so the shift from traditional fertilizer to the organic alternatives that we were using wasn't too bad, and because they made them all themselves, it was kind of an equivocal cost at the end of the day. Most of the cost was like kind of fumbling around trying to figure out how to produce this way, going from a conventional side of things. They had to fire their farm manager, get a new guy in. It was a whole thing. Everybody has to get on board, right? It's a process, yeah. And then this took like 3 years. This wasn't done overnight. This was a lot of small steps to come along to get it where it was. Let's see. So I got to catch back up now. All right. This is other projects we ran in soybean. And I don't know how am I doing on time? Where's my timer?
You've got about 14.
Okay, cool. This is soybean we did in Brazil and in Iowa, where we were able to do mostly just microbial extracts into the soil. So we were running more of like an integrated biological alongside conventional practices. But even just re-stimulating biology surrounding that root system with beneficial biology, we can see like a 5% up to 30% yield increase within the beans. And it has a massive return on investment when you consider the cost of implementing this type of practice. These beans were still continued to run conventionally, but we switched their like herbicide and their like fungicide sprays to as needed as opposed to preventional. So reducing that input while also increasing that output basically at the same time, helping to leverage those resources a little bit more effectively and lower disease pressure. This is a banana project we have in Ecuador. What I mostly want to show here is the difference between these root systems. On the left, we have a traditional treated plant. This is still organic farming. This is an entirely organic farm, but there's been no microorganisms applied to this plant. This plant received a ecosystem transplant, basically. We put beneficial nematodes, protozoa, bacteria, fungi from the region and inoculated this plant with that in a liquid form, and we were able to get massive amounts of fine feeder roots and axillary root growth. We actually did the genetic testing on this, and what we were able to see is a massive increase in hormone production. So you see increases in gibberellins, cytokinins, and auxins, which are all responsible for stimulating that kind of root growth. And that's the role that a lot of these bacteria play within the rhizosphere, is from the outside, sending those signals and hormones into the plant, into the root system, and stimulating that kind of growth. And we all know if you have a bigger root system, your absorption capacity increases, right? You're able to take up more nutrients, you're able to grow a more stable plant structure. And then finally, in Guatemala, we work with a lot of sugarcane. And we were able to work within a extremely conventional system. This is 120,000 acres of sugarcane that they work with. And we worked with them to integrate a lot of different sustainable practices within a conventional system. These guys are still producing on a 7-year cycle. Every 7 years, they rip up their soil, replant the sugarcane. It's 100% sugarcane as far as the eye can see. But we were able to start implementing associative plants. We put nitrogen fixers after they harvested between the rows of sugarcane, which helps to fix nitrogen and outcompete the weeds. We help move them away from burning all of their biomass to harvest into keeping all that mulch on the surface, which increases the water use efficiency. It gets that soil armor built on top of the surface. And we were also able to implement a composting regimen for them. This particular industry has a ton of waste. And we're able to turn all of that waste into a compost that can get applied back into the fields. So that replaced their entire fertilizer program for phosphorus. We just use mycorrhizae now. And this is a place where they produce their own mycorrhizae and they produce their own biologicals to go into the soil. And we're able to see a 3 to 8% yield increase just from a biological program within this intermixed system, where we're able to reduce nitrogen inputs 12% on fields that we were able to identify as optimal reductions. And there was a really great return on the investment of implementing these types of practices. This doesn't have to be really expensive, right? We can produce all of these products cheap and on the farm, and that allows more autonomy to come back to the grower and more profits to come back to the grower at the end of the process. So instead of kind of funneling all that money into fertilizer companies' pockets, we can try to keep a little bit more of that in our pockets effectively. I think we're at the end here. Yeah. Are there any questions? I can jump around slides and stuff. I know it's a lot of info. Yeah.
Do you have any good resources on like companies you like or instructions that you like for creating these on-farm biological like applications?
You can always call us. Yeah, great question. Yeah.
It's you, okay.
Yeah. There's a bunch of them. There were a couple companies out there that were doing similar kind of like on-site microbial stuff or like microbial solutions. We do consultation and on-site production of all of this stuff as well. There are a lot of resources through, if you're very hands-on, things like the Johnson Sewer are great. Learning how to compost, build your own materials, experimenting with that's really good. If you can pick up a microscope, get involved with that, that's fantastic. We use it mostly for like compost and microbial quality stuff. There's a ton of resources. Composting is one of the best ways to grow out native ecosystems. It's organic matter. Microbes live in organic matter. If you build out the right conditions, you can grow out all of these beneficial microbes, and you can inoculate that with different sources of beneficial microbiology from the mountains, from high productive regions of people's farms that aren't super conventional. You can incorporate that in, and that's going to provide a source of inoculation for all of that material. Yeah.
You said use local ecology.
Mm-hmm.
How close? 2 hours? Within 2 hours?
2 hours is fine, like 200 miles is fine. The biggest thing is more of when you consider your biome, right? Like we're up in the mountains, it's high, it's a little more arid. If you were to go 2 hours down the mountain, that's probably not a good idea, right? If you went 4 hours, but you're still in the same ecology, it would be better than going 1 hour all the way down the mountain, right? So try to think of it as what is my soil, what are my conditions, and can I source something from a similar set of conditions? And then as you're doing things, if you're growing that on site, it's gonna be affected by the shifts in temperature, it's gonna be affected by just everything happening within the farm, within your area. But yeah, you wanna try to use things that are kind of locally adjacent to you as much as possible. Yeah.
How exactly did you do the native micro transplant? Was that just like you did an extraction from a native piece and you just liquid extract, or how did that go?
Yeah, we did liquid extracts. So our process that we work with our clients is to put a bioreactor on the farm where we produce all the solutions, and then we extract that into liquid. So it's kind of a classic compost extraction. We don't do a lot of compost tea because once you start adding microbes that grow in soil into water, they're not really in their favorite environment anymore. And once you start adding food to that, there's going to be a couple microbes that win. And so if you aerate that for 24 hours, you're going to create a heavily bacterial solution. It's going to have a couple microbes that are flagellated that do really well, and a bunch of them that don't do great. So if you're doing foliar stuff, that's fine. It'll adhere to the leaf. We usually recommend if you're going to do foliar stuff, try to do like a light micronutrient foliar application because it's much more effective to absorb that from like a chelated micronutrient as opposed to doing like a bunch of. It can get dangerous to do foliar sprays when you don't use all the sugar in the mix and you have a pathogen and then the pathogen uses the sugar. It can get a little complicated. Yeah. Yeah.
How does chelated like iron and stuff like that, does that work well with your extracts? Did you find that impacts your extracts?
Yeah, it can. Chelated is basically going to remove the ion from solution, right? So if it's chelated, it's kind of neutralized. And mostly that helps plant absorption because it can get past, like leaves have an ionic layer that's charged. And so you can get past that outer cuticle and into the leaf structure more effectively. So it just generally helps with absorption of nutrients into the plant. But if you are chelating those nutrients, they're not going to interact with the biology so much, right? Humic acid's a great chelator, amino acids are great chelators, and then there's like a number of other chelators you can kind of use and mix up. Yeah. Was there? Yeah, I think you had a question.
So the difference between an extraction and a tea?
Good question. Extract is taking solid, like a compost material, whatever media you have that's growing the microbes, and you're just knocking them off into liquid, and then you apply them. So you're not trying to change the composition of those microbes at all. You're not adding anything to it or letting it like kind of ferment or grow. You're just taking what's there and putting it in the soil. The reason we do that is because liquid's a lot easier to apply, and it can cover a lot more area. You can take something that's really concentrated and solid and then dilute that into a liquid, and you get much better coverage.
The process is via aeration?
Aeration, agitation, just physically getting microbes off of surfaces. And you can, if you do have a microscope, you can really easily see, yep, I got them off. I can see them in there, right, if you put them under the microscope. And that'll also drive kind of, what am I seeing here, what do I have? And you can kind of base it off of that. Yeah. I think behind you there was also.
I was wondering, how much do you use, so you said you use SAP, how much do you do like foliar nutrition to kind of help with that transition until you get your microbes thriving in the soil?
Yeah, we'll do, it depends on the crop, right, and how frequently they want a sap sample. Usually you're only gonna really do a sap sample if you're gonna spray. So you take that like a week or two before you spray. If you wanna get like a curve of how your plant uses nutrition, you could do it every month throughout a year. But if you already know that, or you don't really feel like you need to do that, it's just a utility basis. So you really want to just pull that analysis. Oh, sorry, I should be repeating questions. Yeah, he asked when you use SAP analysis, like how often you spray micronutrients or do foliars to reinforce the plant. But if you take that analysis, you're going to see if there's deficiencies. You're going to see if nitrates are low or iron's low or zinc's low. And you're going to be able to then say, okay, let's go ahead and make a foliar. A lot of the time we'll do like a super magro mix. We'll make a super magro and we'll do individual nutrients so we can blend them as needed. And then you can go out and spray that. Super magro is just an old, like it's created by a doctorate in Columbia. It's like a method of creating your own fermented micronutrient mix. It uses some mineral fertilizer, replicates what happens in a cow's stomach, makes that all biologically available in a liquid, and you can spray it on your crops basically. A lot of good resources on that too if you look up super magro on the internet. Yeah, super magro. It's SUPERMAGRO. Yeah. Cool. Any other?
Can I get some resources? What books should I be reading so catch up on your vocabulary?
Yeah, we make up a lot of our own vocabulary. You can chat with me sometimes. We have blogs and stuff that we post quite a bit, and we try to like keep them very educational as much as we can. If you do check out our website on stuff, we do a lot of posts on a lot of these topics. You can also read books like, there's a lot of interesting books like Regenerative. There's Teaming with Microbes, yes, thank you. I was trying to pull that one out of my back pocket. The Teaming with Microbes series is really, really good.
I haven't read Teaming and Nutrients yet. They're not as fun.
Yeah. They're not quite as good, but those like types of books. There's a book called Regenerative, which is like really interesting. There's a number of different ones, I would kind of just like Google around and check out like resources like blogs and stuff, and then the Teaming with Microbes series is really great for a fundamental understanding of function of microbes, looking at microbes, understanding those microbes as well. Yeah.
Matt Powers also has a good
Yeah,
series
Matt.
of books.
Yeah, Matt Powers as well. You can go through like resources like Darren Doherty does great stuff on like a whole regenerative chain. A lot of good stuff there too. Yeah.
I guess like I'm also sorry if I'm just taking up.
No.
But specifically thinking about nitrification process and like these chemical processes. Like I know the Jeff Lowell's book goes into that. Is there another thing like another step above that you'd recommend?
If you want to get really technical, you can read the Aldbrich papers. I said if you want to get really technical, you could read the Aldbrich papers. The Aldbrich papers. There's like a 7-part series that covers a lot. They're back in the day. It's from a scientist in like the 40s that was working in USDA. And that covers a lot of nutrient solubilization stuff. It's heavy, and it's horrible to read. It's not great. But also just looking at white papers, if you just go on Google Scholar and you type in whatever you're interested in, there's a ton of resources online. Like all of these papers I'm pulling off, they're just papers I've read over the years that I've been researching nitrogen solubilization processes or whatever it might be. A lot of the time clients will say, hey, I have a question about this, or like I have a problem with this, and we'll go back and dig up a bunch of white papers and do a bunch of research and try to help them figure it out. And so we have just a catalog of like papers that we've been dealing with, and I'll pass them along to the team and to people who are interested. But yeah, white papers are really powerful. They're really solid for education and learning. Yeah.
All right, let's give a round of applause
Okay.
to Miles.
That's right.